Anyway, I'm very happy to introduce our speaker today, who has been with us on one occasion before this, which is very generous. We appreciate that very much. Jonathan Karpf is a biological anthropologist, earning degrees in archaeology and biological anthropology at UC Berkeley. He has been teaching at San Jose State University in the Department of Anthropology since 1987. And we welcome him for what's going to be a great program. Thank you, Jonathan.
Well, thank you. So, first of all, I'd like to apologize. This is a very heavy allergy season, and I have asthma, so I have an asthmatic cough, which I may have to disrupt my own presentation to get through. So this is very different than my previous talk, which was on the cross-cultural construction of gender. This is firmly rooted in biology, which is my field. And so I gave a couple of options to Arthur and the committee, and they decided this would be of interest to the community. So what I'll be covering is the definition of the species that I'll be using in this discussion, because there's multiple definitions of what constitutes a species. I'll be talking about the proximate causes of speciation events, which has to do with reproductive isolating mechanisms, which I'll define further as we go forward. And then I'll talk about different ways of understanding speciation. Two different modes of speciation, anagenesis and cladogenesis, which we'll describe later, and then allopatric versus sympatric speciation. I'll tie one of these forms, cladogenesis, to modes and tempos, sort of how macroevolution occurs. And then lastly, this is leading up to an alternative explanation, an alternative hypothesis to the one that our member James Hunt presented some time ago, as to when that reduction in chromosome number between our closest living relatives, the great apes, who have 48 chromosomes, and ourselves with 46 happened. And I'll be tying it to the origin of the shared-derived character that characterizes our entire family or subfamily, the origin of bipedal locomotion. So a few definitions. Evolution can be defined as a change in gene or allele frequency. Alleles are variant genes in a population over time, just so we start on the same page. And the fact that it's referring to frequencies in a population also gives you an idea of what a population is. Populations are considered the breeding units of species. Unless a species is very small in number and there's a single population, species are comprised of multiple populations, and the majority of breeding happens within the population rather than between populations. So evolution is defined as a change in gene frequency in a population over time, and that also informs you that it's populations that evolve over time, not individuals. Individuals are the unit of selection, but populations are what evolve. Among the forces of evolution, the one that most of you are familiar with is natural selection, co-created by Charles Darwin and Alfred Wallace, which is based on differential reproduction, some individuals having more genes in the next generation than others based on genetic variance. And in terms of this discussion, natural selection is more pronounced as an evolutionary force when the population size is large. In particular, when PE, which is the effective population size, and the effective population size refers to all the individuals who are capable of breeding in the population. You ignore the folks that are too old to reproduce, you ignore the ones that are too young to reproduce, you ignore the ones that are sterile for other reasons. It's the breeding members of the population, and when the effective population size is greater than 500, natural selection is the primary force operating on the population. However, an equally important force of evolution to natural selection is genetic drift, which refers to the random fluctuations in genes and alleles that can happen, and those are more pronounced when the effective population size is lower than 500. And when you consider that for most of our early evolutionary history, human populations were well below an effective population size of 500, you can appreciate that a lot of the variability that exists in humans today is due to genetic drift rather than natural selection. Gene flow is not as important as the other two, but it refers to the transmission of genes between populations. And I'll be talking about gene flow in particular because the proximate causes of speciation are anything that disrupts gene flow between populations. And that's because evolution is primarily divergent. If you have anything that, if this is time on this axis, going this way, if there's any barrier to gene flow within a species composed of populations, such that, so here's the barrier right here, that because the ultimate source of genetic variability is random mutations, mutations that happen on this line, when they aren't unable to exchange genes with this line, will be different than the ones that happen on this line. Oops, sorry. And so over time, the populations will tend to diverge genetically and hence morphologically in shape and appearance, whatever. So evolution is generally divergent. And so we're going to be focusing on those mechanisms that preclude gene flow between populations. So what is a species? Because we're going to be focusing on macroevolution. Microevolution, which most of you are familiar with, is evolution below the species level. We're going to be focusing on macroevolution, which is the appearance of species and higher taxa, families, classes, orders, kingdoms, etc. And so there's not a single correct definition of species. There's multiple ones that have cogency in different applications. So there's genetic species, there's species based on morphology, there's evolutionary species, there's phonetic species, there's cladistic species, etc. There's a number of different definitions of species that are equally valid. But for the purposes of this discussion, we're going to be relying on Ernst Mayr's definition of species, which is referred to as the biological species concept, which again refers to groups of potentially or actually interbreeding populations that naturally interbreed, which are reproductively isolated from other such groups. And that focuses on the population as the breeding unit of species. And then the key to defining whether this individual's member of species A or B is, do they naturally exchange genes? Is there naturally gene flow between those two? So it's based on reproductive isolation. There's problems with this definition, as there are with any, right? So for example, there are species known as ring species. The Arctic herring gull you have populations around the Arctic, A, B, C, D, E, F, G, H, I, etc., circling the Arctic. And there's breeding between adjacent groups of herring gulls. But at the terminal ends, there's no natural breeding, nor even are they capable of breeding. Right? So you have this ring of species, and you get adjacent gene flow between A and B, between B and C, between C and D, etc. But there's no breeding here to here. And then the question is, how many species are there, based on this biological species concept? So there's problems even with extant species with the biological species concept. And clearly, it doesn't apply to paleospecies. If you're an evolutionary biologist like I am, and you're dealing with fossil record, you can't take, this fossil and this fossil and put them on a table and say, well, breed, damn you, can you exchange genes? So it breaks down when you're dealing with paleospecies, but it does have the virtue of being rooted in biology, and it's also directly testable among extant species, and so it's very useful. And so we're going to talk about the proximate mechanisms, and we're talking about reproductive isolating mechanisms, or RIMs, and the first dichotomy is between extrinsic RIMs versus intrinsic. Extrinsic refers to something that's outside the organism, and there's only one extrinsic reproductive isolating mechanism, that's geographical isolation. So for example, if you have a species living in a forest, and then there's a forest fire that burns down the forest and it's replaced by grasslands, which the species can't traverse, the ones that are on this side of the grassland can no longer exchange genes with the ones on this side, and so over time they will become more different because evolution is divergent, right? Another example of this are like squirrels living on the rim of the Grand Canyon. There is a species of squirrel that lives, species A, right here, and species B that is only about a mile away from A. But A is on the north rim of the Grand Canyon, and B is on the south rim of the Grand Canyon. And so that's a geographical barrier, an extrinsic isolating mechanism, which means that A and B will no longer be able to exchange genes, and they differentiate into different species. So that's the extrinsic isolating mechanism. And then I want to talk about two different categories of intrinsic reproductive isolating mechanisms. These are based on genetic variants that are intrinsic to the organism. They're based on ultimately mutational change in the DNA that leads to one or more barriers to reproduction. And among intrinsic reproductive isolating mechanisms, we distinguish between prezygotic versus postzygotic. The zygote, for those of you who's been some time since you've had biology, is the initial diploid cell that's a result of a fusion of a sperm and an egg, right? That's the zygote. And so prezygotic mechanisms occur which make the production of a zygote less likely to happen. They preclude the formation of the zygote. The postzygotic reproductive isolating mechanisms are ones which don't preclude the formation of the zygote, but they kick in after the zygote is formed, and they reduce the likelihood of that individual reaching reproductive adulthood and passing on genes. So that's what I just said just now. Okay, so there are 5 prezygotic reproductive isolating mechanisms, intrinsic ones. The first is gametic isolation. That's when it turns out that there's a lot of biochemistry that goes on in the urogenital tracts of a lot of animals. This is particularly the case in marine organisms that do sort of, mass spawning of dumping sperm in the water, right? You have a lot of species dumping sperm in the water, and that sperm is available to, engage in fertilization with the ova from a number of different species, right? It turns out that also in insects, the vaginal biochemistry is very specific to the species. And so sperm from a male from a different species is immobilized because of biochemical attacks by the flora and the biochemistry of the vagina of the female of a different species, such that you don't get a zygote. So gametic isolation is anything that is based on ultimately biochemistry, which makes the sperm from one species incompatible in engaging in fertilization with the ova from the other species. And this is very widespread among marine organisms and a number of insect species. A second one, if that doesn't work, you have mechanical isolation. This, again, is found in a number of insect species, and this has to do with the morphology of the genital structures. It turns out that the penis of species A and the vagina of the female of species A are fit very carefully like a key into a lock. They're very precise, and so that the male genital of one species can't even articulate with the vagina of the other species because of a different shape, and that precludes the formation of a zygote. Mechanical isolation doesn't have to be on specificity of the genital structures. It could just be on size. I mean, we have this very widespread domestic dog species, Canis familiaris. And using artificial selection, we've bred, everything from, toy poodles and chihuahuas to Great Danes and Irish wolfhounds and Russian wolfhounds. Honestly, how likely is it that a male chihuahua can mate with a female Great Dane? Right? Or for that matter, a male Great Dane and a female chihuahua. Just based on size, there is probably very little gene flow between the extremes of dogs, even though we consider them a single species, right? So that's mechanical isolation. Then there's habitat isolation. That has to do with the fact that close related species may occupy slightly different niches. A good example is Anopheles gambii, one of the primary vectors for falciparum malaria in West Africa. It breeds in sunny still water, whereas a closely related Anopheles mosquito, A. aegypti, breeds in shady still water. Because they have these different habitat preferences, despite being closely related, they're unlikely to exchange genes. There's a number of frog species that have similar habitat preferences. One prefers running water, one prefers still water. And so if there's a habitat preference, which is ultimately based on genetic variability, that's going to reduce the likelihood of genes being shared. And then lastly, as far as prezygotic, there's seasonal isolation. That has to do with the time of the year that breeding happens. Good examples are a toad species, Bufo americanus, which breeds in early summer, while a close related Bufo fowleri breeds in late summer. If you're familiar with the yellow pines up at 4,000 to 6,000 feet in the Sierra, Jeffrey pines, ponderosa pines, very similar species, right? 6-inch cones, although Jeffrey has the spurs pointing in. The needles are the same number, same length. They're plate yellow pines, very close related, but one puts out pollen in the fall, the other puts out pollen in the spring. And as a consequence, they clearly came from a fairly recent common ancestor, but they're not going to collapse into a single species because of this different seasonal mating. And so all of these preclude the formation of a zygote, and they could lead to either incipient speciation, right? Or when speciation happens, it can continue the divergent evolution because the two species now are no longer able to form hybrids and therefore collapse back into a single species. Oh, and then lastly, there's behavioral isolation. And again, this has to do with any behavior that's tied to mating. Frog mating songs vary by species. The blinking pattern of fireflies, which they use to attract mates, varies by species. Among non-human primates, there's different mating behaviors that are very species-specific. And in non-humans, behavior is much more hardwired, right? And so mutations that lead to different mating behaviors can then encourage speciation or lead to speciation just by, a female of species A is not going to be attracted by the mating call of a male of species B, or vice versa. If those don't work, then we get post-zygotic intrinsic reproductive isocyclic mechanisms. You get a zygote, it develops, but then it doesn't live to adulthood. There are a number of closely related tobacco species which remain separate species because of hybrid inviability. They tend to wither and die before setting flowers. They're not as viable as either of the pure species. And so as a consequence, you don't get hybrids between them, and the species stay separate. If the hybrid's viable, and no one would consider mules or hinnies to be non-viable. I'd rather be on a mule than a horse on a very narrow trail, actually. You get a mule if it's a mare and a donkey cross, and a hinny if it's a stallion and a donkey cross. They are perfectly viable. They live to adulthood, but they tend to be sterile. Why? Because horses have 64 chromosomes and donkeys have 62. And so the hybrid has an odd number, which usually isn't going to be fertile. And then if that doesn't work, there are a number of examples of what can be called hybrid hypofecundity. And that's the fact that if you get close related species, sibling species, produce a hybrid, the hybrid individual has a lower, even though they are not actually sterile, they are capable of producing offspring. They do so at a much lower rate than either member of the other two pure species. And in the evolutionary game, it's all about genes in the next generation. And even that slight difference in output can either lead to speciation or keep two species that have already speciated from collapsing into a single species. So it's important to understand that even if we're dealing with evolutionary species, these are the mechanisms that actually lead to speciation. Okay, so as far as modes, there's an older idea which is called anagenic or phyletic or straight line evolution. That's where species A in a wholesale fashion evolves into species B, which then in a wholesale fashion evolves into species C. And so to bring it closer to something that some people here might be familiar with, it's the idea that Australopithecus anamensis as a species evolved into Australopithecus afarensis, which then evolved as a species into A. africanus, and then into Homo rudolfensis, and then Homo ergaster, and then into our species Homo sapiens. This is an older idea, and it doesn't really make sense in any species that has multiple populations or particularly widespread species. And there is actually very little direct empirical evidence that supports this mode of speciation. Cladogenesis comes from the Greek word clade, which means branch, and that's branching evolution. That means that a single line diverges into two different lineages, right? And so species A goes into species A. I mean, it's better to do it like this. Here, let me. You see that you have species A, and then there's a speciation event, and A goes off here, and this is B. And let's say B persists, and it outcompetes A, so A goes extinct, and then B speciates into, B and C, right? And maybe they both survive, and then that speciates again, and this speciates again. It's where you get branches, right? And there's a lot more empirical evidence for cladogenesis. And in fact, what is probably going on for those that still think anagenesis is real is when you're talking about speciation, you have an existing species. The width represents the population size, and then you get a genetic change in one individual, and then it's spread. So when you get a cladogenic speciation event, it's initially a very small number, right? Here's this species going on, and you have a small population that now has this variant. It starts off small, and if it is successful, if it's selected for, it rapidly increases, and as it increases, this diminishes, right? It's out competing, and so what people are seeing with and calling antigenic change is probably something like this. It's where the original species was outcompeted by the successor species, and so it didn't last long enough to be detectable in the fossil record. But there is a school of thought in evolutionary biology, and it's one to which I subscribe, that says speciation is always cladogenic. It's always involving a split between two lineages, no matter how long that each lineage persists, small, short time or a long time. So that's cladogenesis, and again, this is very supported by the evidence because it turns out evolution is a very bushy bush. It's not like a palm tree. It's a series of branches, right? And we know that in long-scale macroevolution, where we get a proliferation of species is by a series of events known as adaptive radiations, right? You get, for example, a proliferation of species when a new niche space is opened up. So during the, we had a reptile species, not dinosaur yet, that managed to make it through the Permian-Triassic extinction event 250 million years ago. 90% of all species on the planet didn't make it through that extinction event, so there was a lot of open niche space. Right? There was this proliferation, adaptive radiation of reptiles, particularly the dinosaur subfamily reptiles. They dominated the Mesozoic, right? And then at the KT extinction event, about 65 million years ago, there went all the dinosaurs. There went about 2/3 of all species. But we had monotreme mammals that evolved in the Triassic, egg-laying mammals. We had marsupial mammals that evolved in the Jurassic, the middle of the Mesozoic. We had placental mammals, our kind of mammals, evolving in the Cretaceous. All three of those lines, at least one member of all three lines, made it through the KT extinction. And if you made it through the KT extinction, where 2/3 of all species no longer existed, after the other side of that, a lot of open niche space. And that's where the Cenozoic has been dominated by an adaptive radiation of mammals, particularly our kind, placental mammals, right? Secondarily, marsupial mammals. And so what adaptive radiation is cladogenesis writ large. It's just a bunch of cladogenic speciation events happening in a relatively short evolutionary period of time. So evolution is very much a bushy bush. So as far as another way of looking at speciation, the older idea, but this is not one which I'm going to discount, I'm just going to talk about the relative times they occur, is allopatric. Allopatric is different place speciation. And allopatric speciation requires first there be the extrinsic isolating mechanism, geographical isolation, right? It's saying that you have members of what were originally a single species geographically isolated from one another by the aforementioned forest fire or a river or whatever, right? A desert, right? And once they're separated by a geographical barrier, then there are one or more of the intrinsic reproductive isolating mechanisms, gametic isolation, habitat isolation, season isolation, that come in place, that even if they find themselves in the same geographical location, they're no longer able to share genes. So allopatric speciation is called different place speciation because it always requires first there to be geographical isolation, followed by intrinsic isolating mechanisms. Sympatric speciation refers to same place speciation. And that's the idea that it's possible to have a genetic event that is large enough in its effect that you could have, especially in a small population, on the periphery of a species range. And this is what I'm going to be tying the origin of bipedality to in a little bit. It's important to understand that if a species is widespread, it consists of multiple populations. And it's also important to understand that the distribution of that species in geographical space is not random. all species have certain demands, right? There's a resource base they need to live. So if here is the species, you're going to tend to have that species occupying three-dimensional space because of its resource base. And unless the resource base is evenly distributed across physical space, which is very rare, it's going to tend to be, the resource will tend to be clumpier in the center of the species range than on the periphery. As a consequence, in the center of the range, you get much larger populations. And as you go farther out from the center, you tend to get smaller populations. And when you get on the very edge of a species range, you have these very small and widely dispersed populations because of the paucity of the resource base. So each of these is a population of a single species. Sympatric speciation is the idea that you could have in, let's say, this small population that's in the periphery of a species range, a genetic event occurring in one individual, right? And a couple of things are operating in small populations that don't operate in large populations. One is, if it's a small population, genetic drift is much more important evolutionary force than is natural selection. In fact, we have empirical evidence that, in a sense, genetic drift in a small population can override selective pressures. You can have something which is actually lower adaptive value increasing in frequency in a small population, whereas in a large population, the one that had the higher adaptive value would swamp it genetically. The other thing that's maximized in small populations, of course, is inbreeding. Right? The small population is a breeding pair of sexually reproducing organisms, right? With offspring. And one thing you have maximized in small populations is inbreeding. Closely related individuals breeding. Hold that thought, we'll come back to it in a little bit. Lastly, as far as macroevolution, there's discussion about the kind of the tempo of it over large expanses of geological time. Again, the older idea, not really the older one, but the one that gained currency early on, is known as phyletic gradualism, because that comes from Charles Darwin's, what he kind of finally fixated on. And it's the idea that you get macroevolution over time by the gradual accumulation of small genetic changes. Right? You get a small genetic change, another one, and over a long period of time, you get enough small genetic changes that accumulate that it leads to enough morphological change in shape, appearance, etc., that you recognize that as a new species. It has an expectation that we should be able to detect this in the fossil record. Right? We should be, if this is actually the primary way that macroevolution happens, we should, if we had a complete enough fossil record, be able to document gradual morphological change over time. We generally aren't able to document that. It's an idea that Darwin came up with, but we have evidence that he originally considered the possibility of large changes causing what we call punctuated change, but he quickly discarded it because the prevailing idea that Darwin was arguing against was special creation by an omnipotent creator, which could make a species out of whole cloth, right? And this idea of kind of very rapid change was too similar to that he discarded the possibility of punctuated change, and he was admonished by his confidant in Darwin's bulldog, Thomas Henry Huxley, that maybe he gave it up too quickly. But this was revised in '73 by Stephen Jay Gould and Niles Eldredge, two evolutionary biologists, and they refer to it as punctuated equilibrium. And this is the idea that, for the most part, in a species history, there's evolutionary stasis. You get no morphological change over time, for long periods of time. And when you get morphological change, it happens in a geological instant, and it happens as part and parcel of a cladogenic speciation event, right? And again, this is also testable, and I could show you slides, I didn't bring them, but we tend to see this much more in the fossil record than we see gradual change. We tend to see species having a certain morphology for a long period of time. I'll do this stratigraphically. Instead of the past being on the left and the present being on the right, the past is going to be on the bottom and the present will be on the top, like we're looking at geological strata. And since my slide is not very good, so if this is, time is on this axis, going in this direction, and morphology, some variable of shape, is on this axis. Gradualism can accommodate both anagenic and cladogenic speciation. I've already told you that there's a school of thought that says anagenesis doesn't exist, and I subscribe to that, but you could have anagenic change, so here's species A, and gradual change is eventually species B, and then you can also have cladogenic change as part of gradualism. Punctuated equilibrium says, what we should see in the fossil record, again, time on this axis, morphology on this axis, is that species show basically long periods of evolutionary stasis. No change in morphology. Oops, morphology. But then at a geological instant, you get a cladogenic speciation event, and now there's a morphological difference between these two. And if this one is more successful than this, then this one will stop and this one will continue. But if this one is more successful, this one will continue. If they're both successful, you can get that. But punctuated equilibrium is the idea that species are characterized by long periods of morphological stability, stasis over time. And then as part and parcel of a cladogenic speciation event, that's where new species come from. And that's where most morphological difference comes from, is from the speciation event itself.
Could I ask you to hold that? Just leave it out like for a second.
Okay.
Step aside a little further.
Yeah. How's that?
Great.
Great. Okay, so having said that, and we're going to actually get this pretty quickly. That's a bad slide. So now we're going to go to actual slides. So I'll turn this off. Okay, so the bottom one is gradualism, right? Small genetic changes over a long period of time, as this thing kind of rolls along, you get new species because of sufficient changes resulting in sufficient morphological change. This hexagonal structure is indicating punctuated equilibrium. There's stability for a long period of time, but when you get enough, then you get a large change and it flips over to another facet, and then that's a new species. That's a new species. That's kind of a visual way to do this. Okay. We have well documented the evolutionary relatedness among our taxonomic order, the primates. And although on the outset I'm using a cladistic definition of our species, I'm using a taxonomy. Taxonomy is a naming system, and there's multiple correct taxonomies. You can focus on morphology, you can focus on evolutionary relatedness and genetics, and you get different taxonomies. They're both correct, they're just focusing on different things. And so by calling habitual bipedal primates hominins rather than hominids, I'm referring to a subfamily level, homininae, as opposed to a family level, a higher level, hominidae. The older taxonomy, which focused on morphology, not genetics, had a superfamily, Hominoidea, which included all the apes and us. Okay? Hominoidea includes the lesser apes, the gibbons and siamang, the Asian great ape, the orangutan, the African great apes, gorillas, chimps, and bonobos, and us. So all of these guys are included in the older taxonomic family, or superfamily, Hominoidea. And I'll use that terminology because I'm going to be focusing on morphology. And so it turns out that superfamily also has evolutionary reality, because it actually constitutes a legitimate clade, an evolutionary clade. A clade is a group of organisms that all share a more recent common ancestor with other members of the clade than any organism outside the clade. And we know that there's a last common ancestor. There was a split between the line leading to the old world monkeys and a line leading to the hominoid primates, the ape primates, about 25 million years ago, right? And all of the lesser apes, gibbons and siamang, and the Asian and African great apes and us are descended from that common ancestor about 25 million years ago, right? And then you had different lines coming off of that. First the lesser apes, then the orang, then the gorilla, and then lastly the human-chimp split. And we know, based on a lot of evidence, that despite them looking more morphologically similar to each other than either to us, that chimps and humans share a more recent common ancestor with each other than either does with the gorilla. Okay? I actually did my doctoral research under an advisor who figured that out back in the 60s. So that split between chimps and us was more or less 5 million years ago. And that's what I'll be focusing on. So despite them looking, gorillas and chimps look more alike, the DNA tells a different picture. Chimp DNA and human DNA is more similar to each other than either is to gorillas. And so this split between the gorilla and this is a line common to chimps, bonobos, and us. This is somewhere around 6 million years ago, 6 to 6.5. Whereas this split between the line leading to chimps and bonobos and the line leading to us is about 1 million years later, is about 5 million years ago. What the shared-derived character in the cladistic sense that all hominoids share is an upper body adaptation. It relates to the ability to hang under a branch when you're moving through the trees, because primates are primarily an arboreal order. Primates, as a rule, live in the trees. Monkeys are good quadrupeds. They walk on top of the branch. Apes don't walk on top of the branch. They hang under the branch. And that led to a number of upper body morphological changes that we refer to as the brachiation complex. Brachiation is arm swinging behavior, right, where you swing under a branch. Quadrupedal monkeys on the ground or in the trees have about this much movement in their upper limbs. Think about your dog or your cat at home, right? If you're walking quadrupedally, you don't need to have a lot of movement at the shoulder, right? All apes, including humans, because we're derived apes, have 360-degree rotation at the shoulder. Why? Because a quadruped has its scapula lateral on the side, which limits the rotation. In hominoids, the scapula, the shoulder blade, was rotated to the back. And that's what opened up the joint, allowing this. Try this at home with your dog or your cat. You know, you'll get bone-on-bone contact and they'll bite you. They're not going to like it, right? Monkeys, same thing, right? Apes can do this, monkeys can't. If you're hanging under the branch, you need complete extension of the elbow. Quadrupedal animals, whether they're a monkey quadruped or a dog or cat quadruped, don't have 180-degree extension. If you're moving like this on the ground or on a branch, there's nothing to gain by having your elbows fixed. They have, try to extend your dog or your cat's elbow. They have a long olecranon process, which impedes that, right? But all apes have complete extension of the elbow. If you're moving like this, look what happens to the wrist, right? Hominoids have increased medial flexion at the wrist. Monkeys can't do this, because if you're going forward on the branch, you need to have that medial flexion. The flip side of having more flexible wrists is it's less stable wrist. That's why the large-bodied apes that are ground-dwelling, like gorillas or chimps who are on the ground, they don't walk with their palms flat like a monkey. They knuckle walk. You've got to lock the bones of the palm with the wrist and the forearm. Otherwise, they would drive their ulna and their radius right through the wrist joint. We're the same way. How do linebackers line up? They don't put their hand flat. They do this. How does a lecturer at a lectern lean? They lean like this, right? Because we have inherently weak wrists because we have increased flexibility. We also have a chest that is wider side to side than front to back. Quadrupedal monkeys and dogs and cats, any quadrupedal animal, it's deep front to back, narrow side to side. If you're hanging under a branch and moving this way, one of the determinants of how far you can travel is how far apart your shoulders are, right? So we have long clavicles, and we have a torso that is wide side to side and narrow front to back. Finally, if you're walking on top of a branch, a tail is very useful in maintaining balance. If you're hanging under your connection point, you don't need a tail. There's a single gene that got rid of the tail in hominoids. Monkeys have tails. Apes and humans don't have tails. We have the gene for a tail, and occasionally a human and a chimp and a gorilla and orang is born with a tail, but the normal condition is to have that gene repressed, and gibbons and siamangs, orangs, gorillas, chimps, and humans do not have tails, right? And so this is a set of morphological features that is shared by all members of that clade. So that's a monkey, that's a human, that's a chimp, that's an orang, that's a gibbon, that's a monkey. And so you can see the wider thorax side to side, long clavicle, scapula on the back, etc. Okay. This is a modern human cranium. That's a modern chimp cranium. This is a cranium from Lucy, an Australopithecus afarensis, discovered in, the Afar Triangle area of northern Ethiopia, dating to about 2.8 million years ago, although the species is older than that. It goes to about 3 million years ago. And craniofacially, if that's all we had, this looks a lot more like the chimp in brain size and morphology than it looks like the human. We would have thought, great, we have an early chimp. It would have been wonderful because we have very few of those. This is a human dental arcade. It's that parabolic shape, right, where the cheek teeth are kind of in proportion with the anterior teeth, the incisors and canines. Canines are not longer than the other teeth. This is a chimp. You've got much larger incisors and canines than cheek teeth, and you have these enlarged canines, and then you have this gap, this diastema, where in this case, the lower canines fit, and you have a similar gap in the lower jaw where the upper canines fit. This is not Lucy, but it's another afarensis, and it's neither the straight-sided U of the apes, nor is it the parabolic arcade of modern humans. It's kind of a, dare I say, intermediate. And this canine is out of its socket, so it's longer than it should. This one's broken off. It has canines that are not as long as any modern ape, but longer than any modern human. And as a consequence, the diastemas are larger than in most humans, although some humans still retain a pretty good diastema, just where apes have it. But it's smaller than the ape. This is a chimp, or it could be a gorilla for that matter, but it's a chimp pelvis. This is a human pelvis. The shared-derived feature of our old terminology, hominid family, or new terminology, subfamily, hominin subfamily, was this lower body adaptation that allowed for habitual bipedalism while on the ground. And we know what led to that. It wasn't a change in the foot, it was a change in the pelvis and then consequently the knee. And it was a rotation of the upper part of the pelvis, the ilium, which in apes is facing front and back. The ilium is like a blade, and there's these three muscles that attach to the back of the ilium. They're the gluteal muscles. Gluteus maximus, we've all heard of. That's not the one that really changed. It's the mid-sized and the small gluteal muscle, gluteus medius and gluteus minimus, that actually changed, reshaping the pelvis. And what happened was, it's clear that there must have been a regulatory gene mutation. Regulatory genes are genes that don't code for a protein themselves or that directly affect the phenotype. They affect the expression of structural genes. There was a mutation in a regulatory gene that controlled the timing and development of the mid-sized and the small gluteal muscles. And the important concept here is that bone is plastic. Bone changes in response to muscle pressure, not the other way around. And what happened is, in apes, which all three of the gluteal muscles act like they're hamstrings, they're muscles for movement, whether they're knuckle walking or whatever, or fist walking if they're an orang. The gluteal muscles are acting just like the hamstrings, moving them forward. But if you're walking bipedally, when I step on my left foot and I lift up my right leg, what keeps my torso from falling over to the right side? I need a countervailing force to keep my torso from falling over, right? That's because of this change in orientation of gluteus medius and minimus. They went from acting as extensors at the hip, keeping this from happening, to acting as abductors. Abduction is when you move a limb away from the body, abduction. And so when I step on my left foot, or let me do this one, I step on my right foot and I swing my left leg, my right gluteus medius and minimus, which are attached to not an ilium that looks like this now, but it was rotated backwards and downwards. So now it's going lateral to the hip socket, right? And it's attached to the top of the femur. Well, it wants to basically abduct my right leg. But if I'm standing on my right foot, it's also attached to my right ilium. It keeps my torso from jackknifing over to the unsupported side. That's the actual change that allowed for bipedal locomotion, was this shift in those two muscles. So there's a chimp pelvis, and you can see that the ilium, so this is the pubis and the ischium, the lower part, but it's the upper part that changed. And basically, the change in the growth of these two muscles brought the ilium backwards and downwards, and that had a number of functional effects. Number one, it brought the center of gravity down and back. A chimp can be bipedal. You give it too many bananas, it can't, knuckle walk. It'll stand bipedally, but because of its ilium and all the muscles attached to it, its center of gravity is about here. So to stand bipedally, it's got to bend its knees, right? What allows us to stand with our knees locked is that the shift of those muscles brought both the bony mass, the ilium, and the muscles backwards and downwards, so that the center of gravity of humans is right where those two lines meet.
Baby got back.
Yeah. Oops, wrong thing. So that's the ilium, that's the modern human ilium. And the other consequence of that, so that's Lucy. That's about 40% of an adult female afferentia skeleton, and we happen to have both the pelvis and the knee, which document these changes. And what we have is right here. So that is a modern human hemipelvis. That's a chimp. That's Lucy. One of the consequences of gluteus medius and minimus rotating this backwards and downwards is that it leads to this notch called the sciatic notch. Apes don't have a sciatic notch on their ilium. We do. That's Lucy. It's broader side to side, narrower top to bottom. Ours is broader side to side, narrower top to bottom. Apes are narrow side to side, broad top to bottom. That's a bipedal pelvis. It also leads to change to the knee. And that's because if you're a chimp, a gorilla, an orang, the femur comes right down to the tibia in a straight line. Because they have a very narrow pelvis, because their ilium are like this, right? One of the consequences of having that change in gluteus medius and minimus, which rotated the ilium down, is it widened the pelvis. That means even in male humans who have hips narrower than females, the femurs angle in towards the knee. There's a femoral angle because you have more efficient bipedalism. If my feet were as far apart as my hips, it's not as efficient bipedalism. You wouldn't have your feet closer to the center of gravity. And that leads to what's called a femoral angle. That's a chimp. That's a modern human. That's A. afarensis. That pelvis is it's not identical to ours, but it's a lot more like ours than it is like a chimp's, and it's documented by the fact that there's a femoral angle. And then because of the femoral angle, there's also a change to the knee that we see in bipeds. These two knobs at the end of the distal end of the femur, the far end of the femur, the femoral condyles in an ape are the same size. In a biped, the towards midline is medial, away from midline is lateral. So that's the lateral condyle, that's the medial condyle of this individual's right femur. That's the medial condyle, lateral condyle of this individual's left femur. Bipeds have a smaller lateral condyle and a bigger medial condyle. Because if instead of going like this, femur to tibia, it's coming in at an angle, what's going to happen? You're going to crush the lateral condyle? No. It gets smaller. The medial one gets bigger to maintain the articulatory surface with the tibia. So if I see the end of a femur, I can tell you if it's a biped or not. Condyles same size, quadruped. Smaller lateral, bigger medial, it's a biped. And that's what we have in afarensis. We also get a change, we get a lateral lip, that's where the patella sits, that's the patellar groove. And in apes, there's no lip to the patellar groove because the anterior compartment muscles, the quadriceps, which go over the kneecap and attach to the top of the tibia. So when I flex my right quadriceps, I extend my lower leg, right? Because it's going across this joint. In a quadruped, the femur and the tibia are in line, so it pulls the patella, the kneecap, slightly upward, right? But if you have a femoral angle, and you flex your quadriceps, you're likely to pull your patella laterally out of the patellar groove. As a consequence, in bipeds, there's a buildup of bone on the lateral side of the patellar groove to keep the patella from being pulled out every time they flex your quadriceps muscles. Afarensis has a lateral lip to the patellar groove. That's the femur of a biped. That's a chimp. That's a modern human. That's Lucy's. Small lateral, big medial. And that just shows you, there's the ilium of in this case, a gorilla, and ours has been brought backwards and downwards, forming that sciatic notch. And there's the center of gravity of a chimp, there's center of gravity in us, and so the knees have to be bent. We have further documentation of the locomotor behavior of the species from a site farther south in Tanzania called Itoli. This is Mary Leakey's group, which discovered a fossilized lava bed. There's Mary Leakey herself, the late Mary Leakey, measuring a track of footprints that was done in, there was a ash fall, a rain which cooled it, and then a number of animals walked across this cooled ash, snakes, baboons, etc., including 2 large-bodied primates where we have no knuckle marks. We have a larger and a smaller one. All we have are the foot marks. And we get a change also in the foot of a biped. Apes have a big toe that's actually more opposable to their other toes than their thumb is to their fingers. But if you're walking on 2 rather than 4 limbs, you need to bring the big toe in line, right? Our weight is borne on our big toe. You could lose your 4 small toes and maintain your big toe, and your gait would be relatively normal. If you kept your 4 small toes and lost your big toe, you would not walk normally because the weight is borne on the big toe, so there was strong selection to get the big toe in line, and that's what happened. 10 minutes, okay, we're gonna take it on home here. Okay, and so there's a reconstruction. So we have bipedality, we clearly went from a knuckle-walking ancestor to a bipedal ancestor, and then this dovetails with James' presentation a year ago. The other thing that we know is this. This is the question that James is trying to answer, and I'm going to propose an alternative hypothesis to James as far as the timing of this event, but it's the same ultimate phenomenon we're talking about. Yeah, I will. Let me just draw this first. So here's us. Here's chimps. Here's bonobos. Here's gorillas. Here's the Asian great ape, the orang. Okay. Orangs have 48 chromosomes, bonobos have 48 chromosomes, chimps have 48 chromosomes, gorillas have 48 chromosomes. The simplest explanation is that this last common ancestor of all great apes and us had 48 chromosomes. It was kept in this line after the orang line split off. It was kept in that line. It was kept in this line. But humans have 46. The question is when that reduction happened and what was involved in that reduction from 40 to 46 chromosomes. James proposed it at the speciation event of Homo sapiens about 200,000 years ago. He then came across evidence that tended to falsify that. I'm going to propose that that change to 46 is part and parcel of that speciation event. That the chromosome reduction is what led to this line being reproductively isolated from this line, which then led to the chimp and bonobo. And we know which chromosomes fused, and that's what you see on the screen right now. Chromosomes, the autosomal chromosomes, the non-sex chromosomes, are numbered based on their length. And it works pretty well until you get to 21, 22. So there's 22 autosomes, 22 pairs of autosomal chromosomes that everybody has in pair, and then females have 2 X chromosomes, males have an X and a Y, the 23rd pair, right? So there's 22 pairs of autosomes, and it turns out the 21st is actually smaller than the 22nd. When they were first doing this in the 50s, they couldn't tell, and they thought 21st was longer than 22nd. Subsequent electron microscopy tells us 21 is the smallest, actually, so they should have been numbered differently. You know that because the only trisomic condition with an autosomal chromosome that lives to adulthood is Down syndrome. That's a third 21st chromosome. If you had a third 22nd chromosome, lethal. Okay, fewer genes to have dosage effect. So in each of these pairs, that's the human number 1, that's the chimp number 1 chromosome. That's the human number 3, that's the chimp number 3. Human number 4, chimp number 4. Human number 5, chimp number 5. Notice how similar those are in the G banding and C banding? That's because we are very close related to chimps. The reason I skip number 2 is, here is the chimp number 2. Here's the human number 2, the second longest. Here is the chimp 13, and here's the chimp number 12 chromosome. These chromosomes are metacentric. The centromere, the constriction point, tends to be in the middle of the chromosome, giving you a P and a Q arm that are roughly the same length. Some chromosomes are said to be acrocentric. The centromere is very close to, for example, these. The centromere is towards one end of the chromosome, giving you a very tiny arm and then a longer arm. Here's the human number 2. Here's the chimp number 13. Look at that. There's the Q arm of the number 2, and here's the chimp number 12. Look. The way we went from having an ancestor with 48 chromosomes to having a descendant with 46 was because of fusion of two. Notice that's acrocentric and that's acrocentric. There's the centromere of the chimp 13, and there's the centromere of the chimp 12. Chromosome fusion normally involves acrocentric chromosomes. You rarely get metacentric chromosomes where the centromere is in the middle fusing. Where you get chromosome fusion is when you have acrocentric chromosomes with a tiny arm and then a much longer arm. Our number 2 is the product of a fusion between the chimp 12 and 13. And what I propose is that fusion event affected regulatory genes controlling the growth of gluteus, medius, and minimus. I don't know this for a fact, but it's testable. I predict that if you sequence the DNA that is around the centromere of the human number 2, you will find regulatory genes that control the timing and development of those two gluteal muscles. Because here's the idea. Mutations are random. You get this fusion event in one individual. We're at the periphery, we're in a tiny population at the periphery of a species range. You get this mutation in one individual. It happens in the sperm or the egg. This fusion happens, and so one offspring inherits, instead of two 12s and two 13s, they inherit, one number 12 from, let's say, dad, a 13 from dad, and from mom, they inherit a fused 12 and 13, right? Now, if this hypothesis is accurate, and I don't know if it is, but it's testable, it's hard to say what kind of gluteal muscles that offspring had, right? They have the original 12 and 13 from one parent saying make gluteal muscles like a chimp's, right? And they have another one that is saying make gluteal muscles which are acting as abductors rather than extensors. And so I don't know how that individual would locomote. I mean, is there a functional intermediate between a quadrupedal knuckle-walking ancestor and a biped? I mean, what is it, a triped? Using 3 limbs? It doesn't make sense. This, by definition, could not have lasted very long. But one thing that's maximized in small populations is inbreeding. All it would take is, say, if that's carried in the female germline, and females produce their eggs before they're born, well, they produce their secondary oocytes to become eggs before they're born. All it would take is producing a daughter and a son, each that had that kind of heterozygous chromosome condition, right? And if there was mating between them, you could get offspring that was now homozygous for the 23 chromosomes. They get the fused chromosome from both parents, right? So what I'm suggesting is this could have taken as few as 2 generations to happen. And if it's happening in a small population on the periphery of a species range, that's going to affect behavior, locomotor behavior. That could be a behavioral isolating mechanism. That could have been at the root of this speciation between the line leading to the chimps and the line leading to us eventually. And so that basically is that is the story. I came in under time. So who has questions? Okay, we need a microphone first. No, wait. They're recording it, so we need to have mics when people are speaking. So a mic is being brought to you right now.
You can repeat the question.
Yeah, well, here you go.
What caused the speciation event?
Is the mic on?
What caused the speciation event?
Well, ultimately it's based on some mutational event, right? So it's a random change in the DNA with chimp-human split. What I'm suggesting, that mutational event was a chromosomal mutation. It wasn't a point mutation. It wasn't some base pairs in the DNA of one chromosome. It was the fusion of the ancestral 12 and 13, creating a single longer, what we now call the number 2 chromosome. And what I'm suggesting is that fusion event altered regulatory genes as part of that fusion event, which then caused a change in the growth of those gluteal muscles, which then meant that individual was born with a pelvis that looked like this rather than like this. And I'm suggesting that first offspring that carried one copy of that fused chromosome, but had the normal ancestral 12 and 13 as well, they would have, it's hard to imagine, they'd have one pelvis that was like a ape's and one that was more like an afarensis, which is pretty close to ours, not identical. And I don't know what that would do for their locomotion. And again, in a large population, that would be selected against. It's clearly not as adaptive as the ancestral condition. But if it's in a small population, genetic drift is much more powerful than selection, right? And because of the inbreeding you get in small populations, all it would take is 2 individuals carrying one copy of that fused chromosome to mate, mother, son, brother, sister. And in 2 generations, you could get an individual who was now homozygous for that fused chromosome. They would now have a functional pelvis because they have both their left and their right gluteal muscles are growing the same way. They would now move through the landscape in a very different way than the ancestral population. And again, if in a small population, that could increase in frequency, right? They have offspring, you get more and more. And over time, you get enough of them that they actually outcompete the ancestral population. I mean, there's far more humans than there are apes in terms of numbers, right? Other questions?
The next question is up here in the front. I'll be bringing the mic around, so just get my attention if you have a question.
Yes, I have a little confusion now. It's perhaps like the problem of the egg and the hen. What is first? You mentioned the function and you mentioned the anatomy, right? What is first? The anatomy change of the bones and then the muscle adapt and the function comes?
Okay.
Or perhaps the mutation that you said lately produces first some change, the bones or the muscles or the function?
It's a good question. What I'm suggesting is that the genetic change that was part and parcel of this fusion event of chromosomes affected the growth of the muscles. As I said, bone is very plastic. It changes in response to muscle pressure. Muscles don't change in response to bone, right? And so if the muscles now are growing in a different orientation, as the ilium develops, it's going to be in the modern human condition, or more like the modern human condition, than like the ancestral ape condition. So the mutational event altered the growth of muscles. And then that, in turn, the growth of muscles altered the bone. Right? And again, mutations are random. It didn't happen because it could lead to bipedality. It happened in a large population of ancestral chimp humans, whatever. It would have been lost by selection. But in a small population, especially if it didn't have to persist for very long, and you can get to a homozygous individual in 2 generations, you get a new way of moving, which offers certain functional abilities that maybe aren't as easy if you're knuckle walking. And it grew in number, and then you got subpopulations of that, and it spread, and that's what I'm suggesting is what led to the hominin line, which then, led to modern humans.
Our time is short, so I want to ask that you keep your questions as short as you can.
Thanks for a great talk. So at least at first, having different numbers of chromosomes was not barrier to reproduction, obviously. Would you comment on that? And
yeah.
Also, are we sure that Neanderthals and Denisovans had the same number as us because their DNA is somewhat fragmentary?
Well, I'll let James respond to the number of chromosomes of Denisovans. Or Neanderthals, for that matter.
Well, Neanderthal, we don't have any real solid hit data, but there was a fingertip found by Svante Pääbo's group. It was sequenced from Denisova cave in the Caucasus. And that Denisovan fingertip, I believe, was dated to approximately 140,000 years ago.
Yeah, more or less.
And it did have the fusion event. They found the place where the chromosomes are stuck together. So this is at least 140,000 years ago.
Yeah, so your initial question was about is a difference in diploid number, say 48, 46, or 64, 62 in the case of a dog and horse, does that mean you can't get a hybrid? No. I can tell you categorically that humans versus chimps and bonobos are more similar in our DNA, are more close related than horses to zebra and then sheep is to goat, right? And we know we are closer genetically than many pairs of sibling species that we know we can get viable hybrids between. So if you're asking me as a thought experiment, if you impregnate a female chimp or bonobo with human sperm, if you get a viable hybrid, given we know, there's no question. Would that individual be fertile? It's unlikely because of the odd number of chromosomes. But viable? Yeah, I know we get hybrids between species that are far more different than we are to chimps and bonobos.
Lions and tigers?
Yeah. Yeah, we're closer to chimps and bonobos than lions are to tigers. You can get hybrids between lions and tigers. So having a difference in diploid number of 2 does not preclude a viable hybrid, but it does preclude us a fertile viable hybrid.
And yet, we're here.
We have time for about 2 more questions. Masuma has the next one.
Hi, can you comment on Homo sapiens spread all over the world, and then there is this extinction you mentioned about those the squatter species. Why there was no speciation because the Homo sapiens didn't mix with each other for many, many years. Right. Why there was no speciation after they moved to different regions of the globe?
That's an excellent question. One is the relatively short evolutionary time that we've been as Homo sapiens. The prevailing evidence suggests that we're the result of a speciation event off of an African Homo ergaster population more or less 200,000 years ago. And that's fairly short, number one. 2 is, for the first 100,000 years of our existence as Homo sapiens, we were confined to sub-Saharan Africa, right? And so there weren't, there was not, you don't get complete gene flow between population in Central Africa and one in Southern Africa, but you get some gene flow, and that maintains the species, right? 100,000 years ago, we start getting some Homo sapiens going out, kind of the Sinai into the Levant area, etc. And then by 40,000, we get them in Europe, and by, by 60,000, we get them in Australia. We start spreading. And so I think it's because there's 2 reasons. One, there hasn't been that much evolutionary time. And the second is the sort of sympatric, large genetic changes that we're talking about here. Once you get a species like ours, the likelihood of such an individual surviving is very low. even if it is capable of surviving, humans regulate their fertility in a variety of ways. I mean, as an identical twin, I can tell you that if I were born in most human cultures, I would not be here. Because the human condition is to have one young. We're designed to have one young. And so if there's twins, either the small one is led to die, and I was the small one of the two, or both are killed or let to die passively because they can disrupt the resource provisioning of the existing kids, right? And so any sort of, I mean, an earlier idea of this punctuated equilibrium model was by an anthropologist named by the name of Goldschmidt in the 40s, and he termed it the hopeful monster hypothesis, right? You get a sufficiently large chromosome mutation that it's kind of like a monster. It's so different morphologically from the parent generation. But among humans, that sort of individual would not be able to reproduce. Even if they're allowed to live, their likelihood of reproducing is basically nil. And those mitigate against an actual speciation event happening in Homo sapiens. We have far more social control of fertility and even in an individual family, let alone on a larger social scale, that the sort of changes that could lead to a new species coming off of Homo sapiens, it's not that it's less likely to happen, but it's less likely to live and to reproduce. And so that nips in the bud the ability of any future speciation events coming from a species like us. Does that make sense?